Hello, my name is Eric Fernandez. I'm project leader at Physiomics, and I would like to thank you all for attending this webinar on the virtual tumor preclinical technology. So the program for today, first we will start with an introduction on our company, Physiomics. Then I will explain the challenges that encompass the modeling of xenograft experiments, then introduce you to our virtual tumor technology, how it works, what calibration data are needed, and finally I will show you two case studies demonstrating how the virtual tumor can help you take the best scientific decision and how it can help reduce costs. So first, let me introduce our company, Physiomics PLC. Physiomics was founded in 2001 in Oxford, in the United Kingdom, and we are based at the Madeleine Center. We focus mainly on oncology. We improve chances of success for oncology candidates by using modeling to, one, predict and understand cancer drug efficacy, determine optimal drug dosing schedules, and find the most potent drug combination therapy and scheduling. We have also new modeling platforms like cardiac toxicity, predictions, population PKPD, and those will be presented in another webinar in the future. Our products and services range from target discovery to clinical trials, and today I'm going to present you the virtual tumor preclinical. We have also some other services like database on drug combinations and regimens, as I said, cardiac toxicity prediction service, and preclinical or population PKPD. Our customers range from large pharma. Eli Lilly was one of our first large pharma customer. We have supported their oncology program since 2007, and they license a customized version of our model player platform. We are working also with other undisclosed large pharmaceutical companies, and we also work with biotech companies, including Cyclacel, Sarum, ICR, CRT, Valirex, and biotechnology services. So let me present you first the challenges behind the modeling of xenograft experiments. As you know, cancer is a multi-scale phenomenon that ranges from a single cell to the whole body. So as a multi-scale phenomenon, it has to be modeled on many levels. Cancer is also a complex multi-scale process. It involves many aspects of biology like cell proliferation, death, invasion, motion, angiogenesis, and all these aspects are dominated by nonlinear dynamics. So to model cancer, you might want to consider diffusible chemicals like oxygen, growth factors, or model the cell colony level, the tumor, with the space occupation, motion, expansion, shrinkage, morphology. And finally, what happens at the subcellular level by using agent-based or ODE models to describe the cell cycle progression for each cell. But first, one has to consider what he wants to model and what he wants to predict. The goal here is really to be predictive. What will happen on xenograft when we apply a drug treatment for single agent or in combination. So the question that has to be asked is how much of that complexity should be part of the model to give the best predictions. Well, at Physiomics, we have studied this question, and we think that modeling complexity and predicting are not the same things. So how complex and detailed should a model be in order to be predictive? It has been shown in many studies that complex models cannot be fully parameterized and are less stable. Indeed, a lot of these models contain a lot of parameters that may or may not be measurable. And even if they are measurable, the level of precision has to be formidable to have a good model. And indeed, it's been demonstrated that simple models are often better for predictions. Let's take, for example, the M3 competition, which is a forecasting competition for mathematicians. And the major conclusion of that competition in 2007 is that simple methods outperformed sophisticated ones. That said, one has to consider the various aspects of cancer therapy to create predictive models. Indeed, to model the tumor response to therapy, we know that cancer therapies involve a vast range of strategies. We can target the cell cycle, interrupt pathways, or damage cell components like the DNA or microtubules, or target the microenvironments. Also, when considering the drug mechanism of action, we have to know the effect of the drugs on the efficacy and the toxicity. And finally, when you model, we want to model drug combinations and consider additivity, synergy, antagonism, and also optimize drug dosing and scheduling by maximizing the efficacy and minimizing toxicity. You have a lot of drugs which work in combination, like gemcitabine and docetaxel, or docetaxel and flavopiridol, where the sequence is important to obtain maximum synergy. One example is docetaxel and flavopiridol, where one sequence is 8 times more potent than the other sequence. So how to predict such kind of aspects of scheduling drug combinations? Well, that's why we have developed virtual tumor. So virtual tumor, shown here in the middle, is a proprietary cell population model, where each cell it contains an instance of a cell cycle model, and that grows in silico, and where we can simulate the effect of drugs and drug combination treatments. To calibrate it, we use cell line growth data, which calibrates the model for specific cell lines. We use also pharmacokinetic time course profiles for each compound. It can be measured in plasma or in tumor. And we use also pharmacodynamic data, like xenograph inhibition data for each drug, or biomarker data of the cell cycle, cell death, and target biomarkers. Using these three data sets, we can calibrate the model for specific drug combination and schedule, and produce simulation, as seen on the right, of various tumor growth inhibition for selected schedules. Here what you see is an overlay of our prediction, which is the red line with the pink area, which is our confidence interval, overlaid with the experiment, where the thick black line is the mean, the average of all experiments. The thin black lines are individual mice data, and the standard error. So I want to emphasize that here it's an overlay between our prediction, which was given beforehand, and the experiment, which was given after. So it's a real prediction, not just a fitting. One case study, in one of a case study, we had to predict 21 different combination dosing schedules that were involving cell cycle inhibitors and standard of care. Out of these 21, 19 predictions were very accurate, as you can see on this figure. So again, our prediction was overlaid with the experiment after we delivered the predictions. So the virtual tumor can be used not only to test hypotheses, but also to design and simulate new experiments, rank dosing schedules, optimize dosing schedules, and check for data consistency. The benefits is that with Physiomics approach and the usage of virtual tumor, you can heavily reduce the number of animal tests. Therefore, you can have a higher chance of success, have an idea of the optimized dose, schedules and combinations. It's cheap and fast. Whereas without virtual tumor, animal testing can be extremely costly, very long, and provides little or no optimization. That's an example of the benefits that you can get in such a case study. In one case scenario, the xenografts experiment extended over a period of 9 months. We demonstrated that using the virtual tumor, the 3 first months of experiments could be used to calibrate the model and be used to predict the other experiments that were done during the later 6 months. So the time saved with virtual tumor was 6 months, around 50K cost saved, and one of the main benefits, of course, is the opportunity to deploy scientists on other projects. This study gave us an award from the NC3R in the United Kingdom. So the data requirements to calibrate the virtual tumor model extend from a minimal data set that can be extracted mainly from literature data to an ideal data set. But the typical data set uses plasma pharmacokinetic profile for each drug in isolation. Then the drug pharmacodynamic data are the proxy, the target biomarkers. Ideally, we can use endpoint biomarkers, which are biomarkers of the cell cycle progression, like histone H3, TOPO2, RB. We use also cell line specifications, like cell cycle phase duration, which can be determined using FACS study or literature data. And finally, we use some limited xenograft tumor growth for control and treated for each drug in isolation. The prediction confidence will extend from OK to best, depending on the quantity and the quality of this dataset. We have used the virtual tumor for a wide range of targets, which are shown on that slide. The nature of agents can be small molecules to biologics, nucleoside analogs, oligopeptides, radiations, target agents, and standard of care. Here you can see an example of drugs and cell lines that we have already used and calibrated in the virtual tumor. So now I will show you 2 case studies where we have optimized from single agent data the combinations of these agents. So the first case study is predicting optimal schedules for a combination of gemcitabine and docetaxel in a breast cancer model. The objective was to use the virtual tumor to improve response in xenograft models using single drug data for xenograft, PK, and cell cycle phase biomarkers. And as I will show you, we have been able to improve a 50% inhibition of tumor growth compared to an original schedule. So we collected plasma and tumor PK for each drug in isolation, then some cycle phase biomarkers, TOPO2 and histone H3, and finally tumor growth for each drug in isolation. We built pharmacokinetic models of both gemcitabine and docetaxel, as shown on this slide, and calibrated it for the data. Then we calibrated the pharmacodynamic part of the model. On these slides, the dashed line are our simulation, and the squares are the experimental data. So we calibrated the virtual tumor for control, docetaxel alone, 1 dose, and gemcitabine alone, 1 dose. And then we use the virtual tumor to try to find optimal schedules. So the strategy was to inject both drugs, gemcitabine and docetaxel, 3 times once a week. And we focused on the offset. So should the gemcitabine be given before or after docetaxel, and how long before or after docetaxel? That's what we ran with our simulation using the virtual tumor. And we used as an endpoint the tumor size at 672 hours, which is the size of a tumor at the 4th week. On the figure, we represent the efficacy of a drug treatment, which is that tumor size. And of course, the better treatment occurs when the tumor size is minimal, and a worse efficacy occurs when the tumor size is maximum. So in order to test for these predictions, we selected these 3 points there and asked our partner to do the experiment. This is a summary of the regimens which were predicted using the virtual tumor. The one on the top, gemcitabine followed by docetaxel with an offset of 4 hours, was predicted to have a worse synergy outcome. On the other hand, we predicted these 2 schedules having better synergy, a blue and the yellow. Gemcitabine followed by docetaxel with an offset of 12 hours, and docetaxel followed by gemcitabine with an offset of 10 hours. The result of these experiments are shown here. The blue curve is the control, the red curve is gemcitabine alone, and the green curve is docetaxel alone. The 3 curves shown below, brown, yellow, and blue, correspond to the schedules I showed you before. So the brown one is the schedule that was predicted to be the worst, and indeed it was no better than docetaxel alone. Yellow and blue were predicted to be better schedules. Yellow was only marginally better. However, the blue schedule was indeed 50% better than the worst schedule, the brown one. So here, the virtual tumor was used not only to predict outcomes, but also used to optimize and select better outcomes. The second case study that I'm going to show you is a combination of gemcitabine and radiation. This modeling was made only with literature data. So we know that gemcitabine and radiation act in synergy in a time-dependent way. This was shown in an article by Milas in 1999, where he showed that gemcitabine potentiates radiation when given 36 hours apart. So can the virtual tumor be used to capture such outcome? We built a pharmacodynamic model of gemcitabine and irradiation, where gemcitabine incorporates and damages the DNA during the S phase. Radiation exposure induces DNA damage during all phases and is given as a single dose at the first step of the simulation. DNA damage leads to arrest and death, cytostatic and a cytotoxic effect. And the DNA damage is in competition with the DNA repair mechanism. So to calibrate the model, we use both in vitro and in vivo data taken from the literature. First, we focused on irradiation data in vitro. We calibrated the model for the effect of radiation, and we compared it with experimental data. In vitro irradiation data can be shown by Varenius in 1998 in the British Journal of Cancer. They exposed cell culture of different cell lines and measured by FACS analysis the cell cycle arrest following radiation exposure. As you can see on this figure, during the next 24 hours, cells in G2M phase accumulate when cells in G1 or S phase are depleted. The profile is different depending on the cell lines. And what we have seen on our simulation is that this was heavily dependent on the cell cycle phase characteristics, especially the doubling time. In the case of a cell line which divides very quickly, like HRT18, which has a doubling time of 16 hours, the accumulation is a lot more rapid than in the case of a slowly doubling cell line like H417, which doubles every 32 hours. Our simulations show exactly the same effect and capture very well the experimental profile of the radiation effect. Gemcitabine calibration was made directly with in vivo data published by Milas in 1999. So on that figure, symbols represent experimental data when solid lines are simulated data. For the red curve is control and the other curves are escalating doses of gemcitabine. Then we calibrated our model for one in vivo irradiation data. We only had one time point for radiation, which is shown on the green curve. When the virtual tumor was calibrated, we used it to simulate the effect of a combination at different administration time intervals. So on the figure here, the green curve is the radiation alone, the red curve is gemcitabine and radiation given at the same time, whereas gemcitabine and radiation given 36 hours apart were indeed had a better efficacy indeed, as shown in the original article. In order to confirm the effect of offsetting gemcitabine and docetaxel, we used our virtual tumor to scan over values of this offset. This is on that figure here, where 0 is when radiation and gemcitabine are given at the same time. And as you can see, the efficacy is lower than when the gemcitabine and radiation are given either 12 hours apart or 36 hours apart in the other direction. Experimentally, it was shown that qualitatively, indeed, it was better to give them either 36 hours apart, or there was also another peak at 12 hours in the other direction. You notice that our model at this stage was giving qualitative predictions because we did not capture the amplitude of this effect. However, this was done with very limited literature data. And that what is important with our model is that we are able to predict accurately efficacy with a limited data set. Indeed, we could improve quantitative predictions later using further model calibration. So, the virtual tumor model already provides a good qualitative prediction of the dependence of efficacy on the treatment schedules for irradiation, and that's extremely useful when designing experiments. That's it for the virtual tumor preclinical. I will just say a word about our the virtual tumor clinical technology, which will be presented in a webinar soon. We have used it. We have adapted our virtual tumor preclinical to a clinical usage to have a translational tool to optimize schedules in the clinic. We have already started its development using around 200 patient data, and currently we are using more development data like we're using tumor images, identifying variable, rim, and structure of tumors. So if you have any further questions about virtual tumor, you can email us or phone us using these details. Thank you very much for your attention, and do not hesitate to ask questions or contact us. Thank you.